Good morning, everyone, and welcome to Emerald Ash Borer University, coming to you live from my office at Michigan State University. My name is Robin Usborne, and along with my colleagues, Anne Marie Nagel from Purdue University and Amy Stone from The Ohio State University, we hope that you will find this webinar informative. Today, we welcome Dr. Deborah McCullough, forest entomologist at Michigan State University and one of the lead researchers in the country on emerald ash borer. Deb has been involved with EAB since its discovery in 2002. Her presentation today will go over what has been learned so far in the Slowing Ash Mortality, or SLAM, pilot project being conducted in Michigan's Upper Peninsula. The goal of the SLAM pilot project is to delay and slow the expansion of ash mortality by reducing populations of the beetle in newly infested sites outside of known EAB infestations. This project is a collaboration between Michigan State University, Michigan Technological University, the USDA Forest Service, the Michigan Department of Agriculture and Rural Development, and the state's conservation districts. If you have questions during the webinar, please feel free to write them in the chat pod. Dr. McCullough will be responding to questions at the end of the presentation to keep the flow of the presentation going. Please stay tuned till the end. Just a minute here, I know I've got that. There we go. Please stay tuned until the end because we would like you to get your feedback, and we'll be providing a link to a survey that we'd like you to participate in. As well, for those of you needing CEUs, your survey information is necessary for us to process those CEUs. The first 10 people to participate will receive an EAB goodie bag. And even if you've received an EAB goodie bag in the past, we'd still appreciate your feedback on this webinar as well. This webinar is being recorded and will be available for viewing this week at www.emeraldashborer.info. You will also find the recordings for all previous EAB University webinars there. And please give us your feedback and tell us a little bit about you. We always want to know how we can make these webinars better. Thank you for attending today. And with that, Deb, welcome, and I'll bring up your presentation and we will begin.
There we go. Hello everyone, this is Deb McCullough. I appreciate those of you who have checked in this morning, and we'll try to get through this and leave some time to answer some follow-up questions. So let's get started today. I am the professor of forest entomology at Michigan State University. I also want to acknowledge Rodrigo Mercader. He was a postdoc here for a couple of years, and he is now down in Kansas at Washburn University, and he has done a lot of work to evaluate some of the data I'm going to show you today. So if we go back, the idea of SLAM got started. It was actually born of frustration, and if you live in an area where emerald ash borer populations are peaking and you're seeing lots and lots of trees dying, you have to think that there must be something that we can do. And in a national sense, eradication was attempted in a number of locations. Minimal success, very little success actually, mostly because it's so difficult to find newly established low-density populations of emerald ash borer. There is still a large-scale national detection survey underway. Some of the states and some municipalities even have their own surveys also underway, but you still have these problems with finding the edge of a population and delineating an infestation or finding a new spot. When a new infestation is found, of course, that county, or in some cases the state, is subject to the quarantine for emerald ash borer, which regulates the transport of all ash material. And other than that, basically you're left to deal with it your own self. And that's true whether you have a front yard with a single ash tree in it, or you're managing a sizable forested area, or a city forester. And we just thought there has to be something better that we can do. And I thought I'd put in a couple of reminders about what happens when you don't take any action. This is an illustration of the area encompassing dead ash trees based on a dendrochronology study that Nate Siegert and some other folks have worked on. And you can see back in 1998, before we even knew about emerald ash borer, we had only a small area of ash mortality, and within 5 or 6 years the beetle had spread, both naturally and because we didn't know about it and people were moving ash around. But the increase in ash mortality was obviously substantial. These photos out of Toledo, Ohio, Dan Herms had the foresight to take these photos, and you can see how quickly in an urban area, a residential area, you go from a very lush, nice, mature canopy to a huge problem for whoever has to deal with those trees. If you look at the economic costs, particularly of landscape trees, because they're the most valuable, if you project where emerald ash borer is likely to be, and we did this beginning back in 2008 and 2009, we projected the distribution of emerald ash borer by 2019, and that's shown here by the gold and the red area. And then we looked at the number of infested or the number of landscape ash trees that were likely to be in that area. And if less than half of the trees were either removed and replaced or treated with an insecticide, the costs are going to be somewhere around $10 to $11 billion during that period. And if we added in the bedroom communities to those urban areas, we're looking at something in the neighborhood of $20 billion just for the landscape ash trees. A large group of us worked for almost 3 years to try to quantify the economic costs of several major non-native invasive forest insects, and emerald ash borer, the economic costs associated with it is greater by orders of magnitude than the other big pests, gypsy moth, hemlock adelgid, sudden oak death, whatever you want to look at. And again, if you look at who's bearing the brunt of these costs, it's primarily local governments. These would be municipalities trying to deal with their ash trees, as well as private homeowners, landowners who are either having to pay to have their trees removed or treated, or are losing value in terms of property values, as well as the timber values. And if you look at ash out in the forest, there's over 8 billion ash trees in the forests in the continental U.S. We don't have good silvicultural recommendations, and really, if you play this out long term, we're looking at the potential demise of ash across North America. And so that basically led us to trying to come up with a strategy to slow the onset and the progression of ash mortality. And SLAM was our acronym. We knew that you had to have an acronym before anybody would pay attention, so that was the one that we chose. And it's very much a collaborative project, and it if you look at the specific goals here, you can slow the rate of ash mortality if you slow the rate of population growth of emerald ash borer. That's what it's all about here. And we do have a number of tools. They're not necessarily all high-tech tools, but we do have some tools that can be integrated into a strategy, and ideally a strategy is designed for a particular site. And in this case with SLAM, we're looking primarily at the ash resource. We know we're not very good at tracking the beetle and keeping up with the beetle, so we're going to focus on what we can work with, and those would be the ash trees. And the tools that we have, as I said, they're not incredibly high-tech. Certainly girdled ash trees are an important tool. People have been girdling trees for probably 200 years, but they're very important in the SLAM project. And a couple of things that we know about girdled ash trees, we've done a number of field studies comparing attraction of adult beetles to girdled trees versus trees that have not been treated in any way, or trees that have been stressed with herbicide. We had trees stressed with wounding and so forth. We've tried to make trees extra attractive with methyl jasmonate, but girdling is just consistently the one treatment that makes trees very attractive. The volatile chemicals that are emitted by these trees are used by the beetles to find a really optimal host. Now, the other point, though, is to recognize that girdled trees are much more attractive than non-girdled trees until the population of ash borer starts to build. And if you look at the x-axis here, this is the density of larvae on non-girdled trees, and this is the ratio of larvae on girdled trees to non-girdled trees. Every one of these points represents a different field site from some study that we've done. And right here you can see that when the densities are very low on non-girdled trees, so you have a low density site, girdled trees are much more attractive. You get anywhere from 5 to 10, even 20 times more larvae on the girdled trees. Over here, right about in here, when you have 20 or 30 larvae per square meter on a tree, you're going to start to see the canopy thinning, maybe some dieback, and at this point the trees in the vicinity are probably all infested. They're all becoming a little bit stressed, and the girdled trees are not going to be so effective. This is just a quick and dirty illustration of a project that we did up north of the Lansing area in a forested area, kind of a swampy forest mix here, and it was a very, very low density population. There's 3 or 4 little golden trees that you can see in each of these areas that were girdled in spring, and then every white spot that you see around here represents a tree that was felled and debarked, and the larvae were counted. And the point here is that having just a couple, actually 4 clusters of girdled trees, where we had 3 or 4 trees that were girdled in each cluster, pulled the population of beetles up into this quadrant. And this area right here represents about 10 acres. 10 acres here, the whole square is about 40 acres. And so the point is that at very low densities, you can actually draw ash borers into a local area, which can be useful when we're doing something like SLAM. And so we use these girdled trees both for detection trees, and we use clusters of 2 or 3 or 4 girdled trees as population sinks. If you're using them as detection trees, you don't want a very large tree. 4 to 8 inches is just about ideal. That's a big enough tree to be attractive to the beetles. You don't have to worry about it breaking in a windstorm, but it's small enough that you can readily girdle it and debark it later in the fall. And the value of these detection trees is that you get not just presence or absence of emerald ash borer, but you can look at the density of the larvae, and you know if the tree is lightly infested or if it's the population starting to build. We can also look at development data. Are most of the larvae overwintering as early instars? If so, we know that's probably getting close to the edge of a population. We also use these sinks basically to absorb the population in a given year. So you girdle 2 or 3 trees, you create a very distinct point source of volatiles. The beetles ideally come to that point source, they lay their eggs on those trees. You take those trees and either debark them or destroy them in some manner and kill the larvae that would have developed on those trees. Systemic insecticides have come a long way since emerald ash borer was very new, and the product that we've used in the SLAM project is Triage. It's applied directly into the trunk of a tree. Like you can see here, there's a couple of different ways. This is called a quick jet, relatively quick way to get imamectin benzoate into the tree. The imamectin benzoate product, this is the active ingredient. It is in the class of chemicals called avermectins, and it's a nice group of chemicals. They're relatively environmentally friendly. They're not hugely toxic to people in the area. They're used, these compounds are used in veterinary medicine as wormers for dogs and horses and so forth. They're used in fish farming. And we know that we can use Triage to protect landscape trees. In fact, I thought I'd put this in because this becomes relevant. If you think about what beetles do throughout their entire lifespan in the summertime, they're going to be feeding on leaves. You can see these little niches taken out of the side of leaves. This is classic emerald ash borer foliage feeding. And we know that females have to feed on leaves for up to 3 weeks before they will even begin laying on eggs. Now, most of the beetles don't travel far, but more likely than not, they're flitting from tree to tree to tree. And if they take a bite from a tree, from a leaf of a tree that's been treated with the imamectin benzoate product, we've done a number of these bioassays, like you see here, where we put a leaf into a petri dish, put anywhere from 3 to 6 beetles in with the leaf, let them feed for up to 4 days. Consistently, we get 100% mortality. They basically take a bite of a leaf from an imamectin benzoate tree and they die. And I think that's very relevant for what we see with the SLAM project as well. In terms of larval density, we have a number of studies. This is the first big scale one that we did. And what we know is that we're getting almost complete control, not just if you treat the trees every year. In this case, you can see these are control trees here, about 85 to 90 larvae per square meter on the untreated controls. These were trees that were treated 2 years in a row with a dinotephuron product or with imidacloprid products, treated 2 years in a row with imamectin, almost no live larvae. More importantly, these are trees that were treated only a single year, and you can see these, this is the control trees here. These are the dinotephuron products. They simply don't last for 2 years. The imamectin benzoate does. We get virtually no live larvae even 2 years later. So we use triage in the SLAM project, and then there's ash utilization. The idea being that if you have a landowner, whether it's private land or state land, and you have some reasonably sized ash trees on there, you can harvest them either for timber, particularly if it's white ash, or at least for firewood. Ideally you use that locally and don't carry it around the state or to other states. So you get some value out of that as a landowner, but more importantly, perhaps you reduce the amount of phloem available for ash borer larvae to develop. And we have an equation now where you can plug in the diameter of the tree, actually in centimeters, but we put it here in inches because most of us tend to think in inches, and you can project how many ash borers are likely to be produced by a given tree. And that enables us to do a lot of things. We can take, for example, an inventory of ash trees for a given area and figure out how many beetles could potentially be produced in that area, or we can do that for trees of different size. And what the point I want to make here is that the data that we have from a number of different forested sites show consistently that there's a lot of small ash trees out in the woods. That's probably not a surprise if you've been out there tromping around, but because they're small, they don't have a lot of phloem and they don't produce very many beetles. Merchantable ash, the trees that are at least 10 inches and potentially could be lumber trees, are not common. That's what you see over here. But even though they're not very common, because they're large and they have a lot of phloem, they can produce a lot of beetles. So the point being that large trees are of interest because they can yield so many adult EAB. We did some modeling projects, simulation modeling kinds of things, where we took an area and we either removed all the trees, we girdled the trees and used them as sink trees, so girdled them and then removed them, or we treated the trees with imamectin. And what we found is, as you might expect, using the insecticide was much more effective actually at slowing population growth than either of the other two treatments. The girdled trees were intermediate. Simply removing ash trees had the least effect on the population growth, and in fact would actually increase spread after a relatively short period of time. Now within SLAM, the idea is to integrate all three of these options. And so getting into what actually happened with SLAM, the inception of this project occurred in the fall of 2007. The Michigan Department of Agriculture had a series of girdled trees they were using for survey purposes across the Upper Peninsula, and they debarked a tree and found that it was identified, and that was the first infestation in the Upper Peninsula at the time. And if you look at the little star here, this is the first tree that was found to be infested. Well, that same fall they were able to fell a number of trees, mostly along roads where they had right-of-way access, and they ended up finding 13 trees that had larvae in them. The infestation had to be relatively recent. There wasn't a lot of dieback or thinning that could be seen, and we've subsequently figured that the infestation there probably began around 2002. Okay, so we had the site at Moran. Then a few weeks later, Andrew Storr's crew from Michigan Tech were working on a research project down here near St. Ignace, which is, if you think of Michigan, this is lower Michigan. There's a bridge that takes you to upper Michigan. They were working in St. Ignace, which is right across the bridge there, and they found an infested tree in one of their study sites. And that was it. And if you look at these areas, these are very sizable areas with a mix. St. Ignace is a little city unto itself. A lot of this area is national forest land. There's private land mixed in. There's rural settings and so forth. And at any rate, we agreed that this would be a good opportunity to try out this SLAM approach. And I think Robin read off the list of cooperators. Everybody listed here, one or more people from these institutions has been very involved with this project. Now, in 2008, the Forest Service had agreed to fund the project in Moran, what we were interested in doing. Late in 2008, if you will remember, the economy was deeply into a recession, and ARAB came about. This was the American Recovery and Reinvestment Act, the stimulus funding. The Forest Service was looking for a project that was shovel ready, remember that term, and they selected this SLAM project to be one of their stimulus projects. So that's how we kind of got into the business of SLAM on a big scale. The project, like I said, it began in 2008. These were the primary activities, a number of different things that I'll talk about a little bit. Today I'm only going to talk about the work that was done in Moran and St. Ignace, what we call the Mackinac. This is Mackinac County in here. This is the Mackinac Project area. I will say there is another SLAM project area near Houghton, and that one is still in progress, and we'll be talking about that probably next year as we get a little further down. I did want to mention, at least briefly, outreach and regulatory activities were a very important part of SLAM. And we had a number of different objectives. The Upper Peninsula had not experienced a lot of the impact of EAB that we saw in other areas, so there was a need to make people aware, not just of EAB, but also the SLAM project, build support for the SLAM project, and so forth. And we ended up putting together this slameab.info website to accomplish a whole lot of objectives at once. Now, survey work, this is where most of the money was spent. The stimulus money, I think we hired over 30 people to work on this survey effort. And we used a combination of girdled ash detection trees, what we're going to call DTs, that were girdled in spring, debarked in fall. They were set up in a grid. We recorded numbers of larvae so that we could calculate density and larval counts per tree, as well as distribution. And then APHIS provided their artificial traps. The purple traps that you see here were hung in trees. And then Bob Hyde led an effort to do an inventory, a very intensive inventory of the ash resource, which we needed for a lot of the modeling work that you'll see in a minute. The trap grids depended on where you were in relation to the origin of the infestation. We had a very dense grid kind of here centered around what we thought was the origin of the infestation, one trap per 40 acres. Out here it went one trap per 160 acres, and then from the 3 to 6 mile radius out here we had a detection tree or an artificial trap for every square mile. These are the results from 2008 for Moran. The green spots here are positive detection trees where we found larvae, so there was 24 of those. Very low density still, around 8 larvae per square meter on average. No evidence of symptomatic trees. 9 of the artificial traps were positive, but in 2008 and 2009 a high proportion of the artificial traps were actually hung on girdled detection trees. So 7 of those 9 traps that were positive were actually on a girdled detection tree that was also infested with larvae. Down in St. Ignace, there were 2 positives recorded. Larvae were in 8 of 20 girdled trees, but the girdled trees were located in 2 of the 40-acre grid cells, one here and one here, and then they were also kind of running a research project, and they got an adult beetle on a sticky band. None of the artificial traps in 2008 captured a beetle. This shows Moran. In St. Ignace we just didn't have a whole lot to work with, but certainly in Moran This is the larval counts, so the number of larvae on each of the girdle detection trees, plus a point for the extra artificial traps that were positive. And the yellow line here is a 800 meter, that's about a half-mile buffer around each of these points. And what we really wanted to do was this. We had a central core infestation that we identified, and we wanted to put in a lot of clusters of girdled trees, with the idea being that these trees were beginning to get infested. We would retain the beetles in the center of the core here, and then in the perimeter or around these core trees, we would inject as many trees as we could afford with the triage product. Therefore, if a beetle got away from the girdled tree clusters, chances were good they would hit one of these treated trees around this buffer. Out here where we had kind of isolated trees that were turned out to be positive, we called them satellites. We were going to girdle one or two trees around the tree that had been positive the previous year, and then again infest trees as best we could in the vicinity. That was a great idea, and I think it would have worked very well, and that's not what we ended up doing, and a lot of it had to do with just the scale and trying to ramp up from a relatively small scale project that we started with in 2008 up to the ARA project. And we probably picked about the worst possible place to try doing this SLAM area. This is the Mackinac Project area here, and the Hiawatha Forest was heavily involved in the initial development of SLAM. And then it turned out that they weren't going to be able to allow us to treat any trees, and in some cases we were not allowed to girdle trees on national forest land, which encompassed a lot of the area. Fish and Wildlife had an endangered dragonfly that was initially had designated habitat right in here, and in 2009 they expanded it to this entire yellow area where we really couldn't take much action. There was a wild and scenic river corridor, there was a wilderness area, there was a research area. In all cases we were prohibited from doing at least the insecticide treatment, and in some cases the girdled trees. There was logistical things. The state had to improve the contract to pay the survey crews. Sometimes you couldn't find the landowner. A lot of non-resident landowners, we needed to get access to private property and so forth. And as opposed to what we had planned, this is what we ended up doing. These if you look over here, we did get some sinks, some girdled clusters of trees in the center of the core like we wanted. A couple down here. I think this was a cemetery down by St. Ignace. These are trees that were injected with triage, mostly along a couple of roads here. And then the MTU folks were able to inject a lot of trees up and down. A lot of these were landscape trees here that run right through the middle of St. Ignace. And I think this was a cemetery and a park down here. 2010, kind of similar. Again, some more trees were injected. I think a total of 500 and some trees were treated in 2009 and 2010 in total, counting Moran plus St. Ignace down here. And then we got some more sinks or high density girdled trees, high density detection trees, which is what the Forest Service preferred that we call them, where we had 2 or 3 girdled trees in a couple of these areas. This bordered the wild and scenic river area. We couldn't take any action up here, but we tried to create this more or less boundary of girdled trees here. Same thing down here, and then a few more sinks in this area. What we did to evaluate it, this is a really tough thing to evaluate because you don't have a control, and we have to use simulation modeling and try to compare what we observe in terms of population density and population growth as well as distribution with what would be expected if we had taken no action. And this is the approach that we're using for the evaluation by and large. And it starts with this coupled map lattice model, and bear with me a little bit here. A lattice, if you think of a lattice, it's just a grid, and it's coupled because we have one grid for emerald ash borer, we have one grid for ash phloem. And what we do is we can start with a population of emerald ash borer, and we track the larvae that are going to develop in a single year. Those would be larvae overwintering as fourth instars or prepupae. And we track the 2-year larvae. You can see that's in red here. Those are very lightly infested trees, and when the trees are debarked, those would be larvae overwintering as early instars. And of course, as the population grows, over here we have ash phloem, and those larvae are going to start eating ash phloem, and at some point they will eat all the phloem in a given cell. There's a number of different parts that go into this model, and I give credit to Sandy Leopold, who helped us build this initially. We have adults that emerge every spring, and again we track the 1- and 2-year cohorts. Those adults disperse. We have a dispersal function built into the model that's based on a number of field sites, and it has been modified based on the data coming out of Moran. And then the population grows over time, and as I said, the larvae eat phloem, and this is all based on empirical data from a number of different sites where we've done projects throughout Michigan. The dispersal function is kind of a key aspect of this model. Again, it's derived from field data. We do have a bias towards girdled trees. That's what we're trying to show right here. But as I said before, as the trees become more and more infested, we reduce this bias because all the trees at some point are stressed and attractive to the beetles. So here is what it looks like over time. You start with an initial ash borer population. It grows, it disperses. You can see, get my little arrow back, you can see down here the population building up over time. If you start with ash phloem, and we can be very spatially explicit, if we know that ash phloem is in a given cell, we know the number of trees, we know the size of the trees, we can calculate how much ash phloem is in a given cell. And that's what populates this side of the model. And over time, as the population of ash borers builds up, the amount of ash phloem in the area is going to decline. So ash was inventoried, a number of plots both in 2010, more plots in 2011. This is what the ash phloem density looks like if you map it across the project area. And these are the data in terms of the ash borer survey. So this is 2010. These are positive detection trees and artificial traps. This gives you the larval counts more or less. The darker colors mean the trees were more heavily infested, kind of here in the center. 2011, you can see we're getting spread. We've got ash borer out to the edges of these trapping area, and you can see more larvae on trees in some of the expanding here in 2011. Now, to analyze this, I couldn't think of any concise, simple bullet point way to explain how we analyzed it. If you're interested in this, I can talk to you, I can send you a report that we wrote and so forth. But one of our big questions was whether or not the triage injections, what we were able to do in the Mackinac area, or the sinks, were able to affect the population growth of ash borer up in this area. And we basically are using statistical methods for this, a generalized linear mix model. A couple of key points is that we assumed, we worked with data from the detection trees, so we worked with the larval counts. We assumed that the number of larvae in a given detection tree might be influenced by the treatments that took place within about an 800-meter radius. We know most larvae stay within 100 meters of the tree they emerge from as adults. They tend not to travel very far if they don't have to, but we also know they will travel up to 750 or 800 meters. Some portion of them do, and so we weighted this by distance. Did some other kinds of things here. I don't want to spend too much time on this because I know people's eyes will start to glass over. The other question is looking at spread, and we had this incredibly intensive data on ash borer. We'll never get this kind of data again, I'm sure, in terms of where ash borer was over a period of 3 or 4 years, and we looked at whether the insecticide injections or the sinks were able to affect spread. And we do this with both a maximum likelihood approach, where in this case we're looking at the probability that a given detection tree would be detected as positives based on the other traps that were within 2 kilometers, 2,000 meters here, because if you look at the flight mill data and some of the data we've got from field studies, it's very likely that a small portion of beetles, particularly female beetles, are able to fly at least a mile. We also have a model. As I said, we've worked with this dispersal function, and we looked at how well our model was able to predict where ash borer turned out to be. So let's get on with it. Key results, instead of going through a whole lot of details, we know that even though we weren't able to inject trees where we wanted to inject them in the spatial distribution that we would have liked, and even though we were able to inject only a very small portion of the ash trees up there, we did have a significant and very detectable effect on the population growth of ash borer. And a couple of key points. The trees that were injected in 2010 slowed ash borer population growth the following year in 2011. Trees injected in 2009 slowed the growth both in 2010 and in 2011. So the effect persisted for 2 years, which is what we had kind of expected. The number of treated trees was significant. The area of treated phloem was not significant. We tested those separately. And we were not able to detect any effect of the insecticide treatment on spread. So population growth was slowed by the insecticide, but we either didn't treat enough trees or the insecticide simply does not affect the spread. Now the sinks, we analyzed these separately, and the results were a little bit more complicated. Sinks obviously have an effect for only 1 year. You girdle the trees, you let the beetles lay eggs in those trees, then you remove the trees. You debark them completely, it kills all the larvae. The number of sinks did reduce population growth the following year. The area of phloem in the sink trees was also significant, but it was a very local effect. So as you moved away from a given detection tree, the effect diminished, and all the interactions were significant. And what it basically means is that the sinks, the girdled trees, 2 or 3 or 4 girdled trees, attract the beetles that are in the local area. And so you get oviposition on your girdled trees, but also on the nearby trees, kind of a spillover effect, which can be a good thing or a bad thing depending on what you're trying to do. We also found, again, that the sinks had a small but detectable, statistically significant effect on spread. They actually reduced spread by, again, more or less consolidating the local ash borers in the area. One of the things that we found is that effect depends on what proportion of phloem in the area is actually girdled and used as sinks. And so what you find is that if there are not very many ash trees in a given cell, which is how we analyze this, if there's not very many ash trees, a lot of beetles disperse and you don't have a strong effect on slowing spread. If you can girdle a high proportion of the ash trees, then you're going to retain more of the beetles. A lot of that's just common sense. It's just trying to figure out how to analyze it, I guess. We also went through some statistical, rigorous and analytical methods to figure out the rate of spread based on where we think the infestation started in Moran as well as St. Ignace. And this is basically what we calculated, somewhere between 1,500 and 1,750 meters per year, so that's roughly a mile per year in terms of increased spread, and that's based on the slope of this line here. St. Ignace was a much smaller infestation when it was discovered. Not only was it smaller, I think it was probably more recent for a lot of different reasons, and you can see the spread rate is considerably lower there at St. Ignace. We looked at our model. In this case, the black dots here is the distribution of ash borer that we actually observed in Moran in 2010, and over here in 2011. This is 2010, the observed distribution. This is 2011, the observed distribution. This is the distance here from the origin. So you can see out here we're out around 8,000, 10,000, 12,000 meters from the origin. This is about 6 miles in here. And the gray points here, the light colored points are our original simulation model. The gray colored points are after we tweaked the dispersal function or accounted for the effects of sinks and insecticide trees. And you can see we have a good fit in 2010 out until we get out here in 2011. Our model fits very well again out until we get 6, 7, 8 miles away. And what that represents is two things. One is our model is pretty good. We can predict how beetles are spreading pretty well, except out near the edges. And that's because the density is very low. Only a few of the detection trees are infested, and we don't have very strong prediction abilities or detection abilities out there. So, if trying to summarize what we've learned in a coherent manner, this should actually say not treat, that should be treat. Treat as many trees as possible with triage to slow EAB population growth. Over and over again, we're seeing that this is the most effective way to protect the trees, retain the trees. You keep all the values, the stormwater capture, the shade, everything else, whether you're in a forested area or an urban area. You know, you can get away with treating trees only once every 3 years and just do yourself a world of good. Now, the SLAM project has shown us that it's more important to treat as many trees as you can rather than trying to select out some large trees or to treat just a few trees in the center of an infestation. Distribute your treatment, treat as many trees as you possibly can. Now, we could not find an effect of the insecticide treatment on spread, and that may be because we treated only a few trees. It's almost certainly because the trees that were treated were pretty much clustered over where there was private property or trees along a right-of-way and so forth. But it also shows that we really don't know a whole lot about long-distance dispersal of ash borer and why it happens and what triggers it. In terms of removing trees, if you have heavily infested trees, there's no reason not to remove them. If they're pumping out a lot of adult beetles, it's best to take them down. You can get some value for the landowners there. We actually got a timber sale set up in the Moran area. Some white ash, a lot of maple and so forth, but the landowners got some value out of it. And those big trees, the ones that are going to be valuable in terms of timber, are the trees that are going to produce at least half, if not more, of the ash borer in a given area. It's not enough to remove trees alone, though. We're just seeing this over and over again, both in terms of the simulation modeling as well as in real life out in the field. You need to combine this ash removal and the phloem reduction with some other strategy. Otherwise, you will eventually remove the trees and you're actually going to increase the rate of spread, and you need to have some effect on population growth, either with the insecticide or with the girdled trees. The girdled trees are scientifically, I think they're very interesting. It's something that's difficult to do. I realize in an urban setting it's going to be more appropriate in rural or forested settings, but you know, there's opportunities for this. You have trees along roadsides. Sometimes there's ash trees that are growing through wires that need to be removed for some reason, and if you girdle them, they can be very effective in terms of congregating the beetles to a point where you can get rid of them and slowing population growth as well as spread. If you're going to use trees for detection trees, I know a number of people are setting detection trees this time of year. You don't need anything bigger than 8 inches, and really a 6-inch tree, 4, 5, 6 inches is about ideal. Make sure you either debark them or you destroy them some way to prevent those larvae from emerging. Otherwise, you're just breeding beetles. And as I said, you can attract beetles, keep them in a local area, and help to slow the population growth. And I think one of the really key points is that combining sinks with the insecticide treatment, in other words, what we originally envisioned, where you have sinks in the center of an infestation surrounded by trees that are treated with the triage product, is probably going to be a very effective tactic, coupling the sinks and the insecticide treatment. So in terms of what we're going to try to do next, as I said, there's a whole nother site up at Houghton that the Michigan Tech people have been working on since 2009. They have done a lot of insecticide treatments, some girdling, some tree removal. They've been releasing parasitoids there. We expect to get that data this summer. Rodrigo will be working with me to get that analyzed. We also would like to use our simulation models, the different components that go into these models, not just to analyze the Houghton data, but also to simplify them and develop a more generalized model that maybe other people could use. In other words, if you have a spot of ash borer, it would be nice to be able to come up with a good estimate of how fast that's likely to spread and where ash borer is likely to be, and that's what we're going to see if we can do that with some degree of confidence. One of the things that we haven't done yet is to use the data from forest health plots. There were 111 forest health plots established throughout this Moran area. In here, the data came in over the winter, and we'll be able to use those to see what's happening to the ash resource. And ideally, perhaps we can predict whether the treatments that we were able to take in Moran and St. Ignace and probably more importantly in Houghton actually are protecting the ash resource over time. So trying to summarize here, I think that what we're going to get out of SLAM, and again, this is a work in progress. You know, you're trying to slow the population growth, slow the rate of ash mortality. It's going to take some time to evaluate this and to see what we're able to accomplish. But it's certainly something that can be done not just in forested areas, but in urban areas, residential areas, neighborhoods, and so forth. You're buying time. You're avoiding this 3 to 4 year period that we've seen so often, where if you take no action, a few trees become infested, a few more trees become infested, and suddenly you're in this horrible situation where all the ash trees are declining and dying at one time, what you'd call a catastrophic situation. That's not good for anybody. Taking an area-wide approach yields greater benefits. I think the sum is greater than the different pieces. One of the things we found is that when you have an area-wide approach, you get estimates for treating trees with insecticides that are lower than they would be if individual people were having trees treated. As I said, you can set up viable timber sales if you're in an area where that's an option and so forth. There's also been a number of spin-offs that have come out of the project so far. We've looked at you doing SLAM in an urban setting, a publication there where we looked at the costs and benefits of treating 10, 20, up to 50% of the trees with triage annually, and showed that economically it makes huge amounts of sense to do a lot of treatment. And we're also in the process of looking at the detection trees and the artificial traps and their ability to detect low density infestations of ash borer. So with that, I'd just kind of like to acknowledge there's been many people that have participated in this project to one degree or another. The SLAM Management Board has been and continues to be very active with the project, and then other people, Rodrigo, Gabriel, who makes our maps, came up with a lot of the GIS techniques we use, a number of other people there, and again the funding that came from the Forest Service got us started and is keeping the evaluation work and the work at Houghton underway. That's being funded through the Forest Service, as well as the stimulus money and APHIS, who provided those traps. So that's all I have. If anybody has questions, this would be a great time. Otherwise, thank you very much for your attention.
Thank you very much, Deb. Looks like Don has a question, maybe more of a comment there for you, as far as doing a mini SLAM in the Allegheny Park in Pennsylvania. Can you read that? Can you see it there on your screen?
Yep, I'm reading it right now. So I assume everybody can see this. If not, Don is saying that within Allegheny Park they removed ash trees that were destined to become hazard trees, treated trees in an area that the park wanted to protect, and also released some of the Asian parasitoids. I think and developed a plan for urban communities, parks, forested stands. I think it's a great idea to protect some ash seed sources. I know there's some groups in Canada that have identified areas where they're trying to treat trees just to save seed sources, so that if and when we get a little bit better at dealing with ash borer, there's still a seed source there. It looks like a really nice plan that you have there. The one thing I might mention is that in a situation where, such as Don's, where you have trees that are becoming hazard trees or could potentially become hazard trees, there's an opportunity sometimes to girdle those trees in the spring, let them serve as attractants for the ash borer during the summer, and then take them out during the winter. Just something to think about. So I wish you all the luck in the world there, Don. It looks like there's a question from John. In terms of having lots of small trees in the understory, I actually don't think that removing those trees is going to have any positive impact on spread, to be perfectly honest. Number one, it's a lot of work. Number two, as you remove ash trees, when the beetles can't find an ash tree, they will fly until they do find an ash tree. And so I don't think that's going to buy you anything there. What I would say is, if it's an area where you can treat some trees, maybe you can girdle some of the younger trees that people are less likely to be concerned about, treat some of the trees in the area. Otherwise, if you're going to try to remove some trees, at least girdle them and get some use out of them as a sink tree before you take them out. But I don't know that you can expect a huge impact on spread unless you get really ambitious with the girdling. And Don Egan is typing in obnoxious phrases like "go blue" and yeah, "go green." There you go.
Sometimes it's just kind of hard to weed out the riffraff in these things. But it looks like there are more people typing, so we may have some more. Just hold tight for a second.
It's good that Don has a sound ash borer plan underway. I think that he must have put more thought into that than into his preferences for sports teams. Dottie is asking, what is in the artificial traps? The artificial traps, the big purple traps that are hung in the canopy of the trees, those are traps that are distributed by APHIS typically to state cooperators. And they're purple because the beetles will respond visually to certain shades of purple and certain shades of dark green. They are sticky, and the beetles that fly to the traps are just caught. They're physically trapped in the sticky material. And there is a lure on the traps. There's actually one compound that is produced by ash leaves. It's called cis-3-hexanol. And another compound, typically they use manuka oil, which is derived from a tree in New Zealand, the New Zealand tea tree. But the lure, the chemicals that are used in the lure, are very similar to those produced by ash wood and bark. And so they are host volatile based lures designed to attract the beetles to the sticky traps. They're not toxic. That's one of the good things. You can put them up there and not have to worry about any kind of toxicity issues. Let's see, Don. In urban areas, what are your thoughts regarding treatment of smaller ash trees versus removing these and just treating the larger trees? Boy, I would say if it's a municipal trees, I actually, depends on what you want to do. The larger trees obviously have more value in a lot of different ways, different senses of the word. I do think, though, that one of the things we're seeing with the SLAM project is that treating as many trees as possible is going to have a stronger effect on slowing the growth of the overall population as opposed to just treating a few trees. Now, granted, once you remove the smaller ash trees, you can focus your effort on treating larger ash trees. That's an option if you want to start taking the small trees out. If you're going to do that, I would do it over time and develop a strategy so that at any given time you have a proportion, at least 20-25% of the trees have been treated in the past 3 years, so that you are increasing the chances that a beetle is going to hit a toxic tree and basically die. So in terms of do you leave the small trees, do you treat the small trees, there's a lot of other thoughts, a lot of other factors that go into those kinds of decisions. If you're simply trying to slow the ash borer population, I would just treat as many trees as you possibly can. And the small trees can usually be treated more efficiently than the larger trees. But if what you're trying to do is to be efficient with your resources, if you have the opportunity to replace those small trees now, and you might not later, that may be something you want to do. And again, if you're going to start a replacement project, girdle the trees for a year, get a year's worth of use out of them as sink trees. Dan has a question. Does a beetle only breed and lay eggs once? The females can mate with males multiple times, and the females actually lay eggs for several days, maybe as long as a couple of weeks if they live that long. They don't lay their eggs all at one time. They lay sometimes a single egg, sometimes 2 or 3 or 4 eggs will be laid in a row in a bark crack. Then the beetle will go feed on ash leaves, rest. The males are always looking for females, and if they get a chance to mate, they will. Eventually the female will come back and lay a few more eggs. We've had females in the lab that have lived for over 6 weeks and have laid over 250 eggs. But I don't think that happens very much in real life. I think mostly they maybe get 30, 40, 50 eggs laid before they die. Don's providing some nice links to the program that Ho-Ping Lu developed. Again, it's a multifaceted program that the state of Pennsylvania is using, and that'd be something that'd probably be of interest to people.
Okay, folks, we've got about 4 minutes, so if anyone else has questions, please, can you please get those typed into the chat pod? Or, Dr. McCullough, can we put your email address on, just in case they have questions that they think of afterward?
Sure, that'd be fine. You want me to read it, or are you going to type it in there?
I use it a lot.
Okay.
There you go.
The note shows that multiple attendees are typing. Dottie had a question about what about sterilization of male beetles. It's an interesting idea. As far as I know, it's never been tried. I know that's been done with Mediterranean fruit fly. They tried it with gypsy moth a time or two. Maybe something that comes about in the future. I know in some cases with some insects, the sterilized males don't compete well with the wild males, so that seems to be a bottleneck sometimes. But who knows? 10 years from now, we may be talking about male sterilization. Let's see, John. Is there a thought that if we can slow the spread to some degree, maybe we'll come up with a more permanent solution in the near future? Boy, that'd be awesome, wouldn't it? I think there's a lot of advantages, not just to slowing the spread, but slowing population growth. I think if you look at what the city of Milwaukee has done, for example, they're on a very extensive treatment program where they're treating their city trees with the triage product, and it's bought them years. They don't have ash borer killing trees. They can do a planned, they call it a staged transition, where over time they're going to replace their ash trees with other species and diversify their urban tree planting, but they don't have to do it right now. And that's one of the advantages of buying time with some of these strategies. I think you're right too, John. Research people are still working on ash borer, and a lot of research people are working on ash borer, and I certainly have hopes that we're going to get better tactics, better options, better tools for dealing with ash borer. And so again, anything that you can do to buy time is going to be beneficial one way or another. Looks like, do you see triage being labeled for a 3-year treatment regime? That's going to be up to the company, I believe, that manufactures and distributes the triage product. I think there's some question if you have very large trees, there's some idea that you may still want to treat every 2 years if your idea is to protect that very large tree. But certainly the trees that most of us would be dealing with, things that are 12, 14, 20 inches and smaller in diameter, could be treated every 3 years. The EPA labeling process is interesting. Let's just say that. So whether the company pursues that or not, I'm not sure. Andrea, how are you? If a company can only treat with an imidacloprid pesticide, is it worthwhile? And if so, how would you change the procedure? I think in that case, if you're treating with imidacloprid, you're not going to get the same level of control, and you're not going to get the multi-year control. So then there's an emphasis on treating as many trees, as great a proportion of trees as you can. And I think, yeah, anything that you can do to get those trees treated, a combination of treating trees on public land, encouraging people to treat trees on private land, and trying to do it annually, is going to be a benefit.
I see Donald had one question about host resistance. Dan Herms at OSU.
Oh, there we go. Yep. Yep, sorry, Don, I skipped that question. Host resistant, yes, there's a big group at Ohio State. Jennifer Koch with the Forest Service and a number of people, Don Cipollini, who's at another university, are all working on trying to figure out host resistance, in some cases at the subcellular level, looking at proteomics and enzymes and so forth. Lingering ash is the term applied to ash trees that are alive in areas where you would have expected them to be dead. And sometimes you go into an area and you see 5 or 6 trees that are certainly big enough to have been colonized and killed, and yet they're still alive. And I think it's good to track those trees. I know they're working on some of them, and they're finding that even when you expose them to ash borer in a laboratory situation, they are somewhat resistant, certainly more resistant than some of the other trees. So Don, as far as your question, locating lingering ash, for me, not so much. We don't have funding to do a whole lot of that, but certainly I would encourage you to put a tag down at the base of the tree and take GPS coordinates so that you can check those trees out, return to them. If vegetative propagation techniques get worked out and become a practical, viable option, you may want to start to propagate some material from those lingering ash trees. So I think it'd be a great idea to keep an eye on them. Sometimes those trees are just trees that got lucky and escaped, and a couple years later they get infested and die. But you got to believe there's a little bit, some trees are always going to be a little bit more resistant than others. So I would certainly encourage anybody to track them if they can.
All right. It's now 2 minutes past the hour. I think if anyone has other questions, why don't you try emailing Deb? And I want to thank you, Deb, again for all the good information. We had a lot of folks that were very interested. Appreciate the time you've taken to be on Emerald Ash Borer University, and I am going to be closing the meeting here in another minute or so. So thanks again for participating, everybody. I hope you got as much out of it as I did. I think it's very interesting. Thanks, Deb.
Thank you. I saw some old friends on the list of people attending, so hello everybody. Hope you're having a great week. Thanks a lot.